So, got it. Yes. Okay. Thank you for the kind introduction. So I'm here today to talk to you a little bit about EPR application in biology. Let me start with share my screen if I can. I think I can. Okay. So to tell you a little bit more about using Expand CV EPR to probe biomolecular structure and function, I want to start with a very abbreviated, very simplified introduction to EPR. So in EPR, we look at molecules, ions, and atoms that have unpaired electrons. Because they have unpaired electrons, they have an electronic spin larger than 0, and they have a magnetic moment. So for our biological applications, that means we need an intrinsic paramagnetic center, a radical. And that can either be, let me see if I can find my laser pointer. This can either be an organic radical, either an amino acid radical or a bound cofactor amino acid radical. For example, tyrosine radicals are involved in photosystem 2 activity. Or it can be a metal center. So usually it's a transition state metal at a EPR active oxidation state. Heme clusters, iron sulfur clusters, magnesium, manganese or chrome ATP complexes. But if you don't have any indigenous paramagnetic center, you can still use it by simply using site-directed spin labeling. So in site-directed spin labeling, a spin probe is attached to a specific position in your system. It can be a protein, nucleic acid, lipid, nucleotides. In the most common application, it's a protein. The mostly used spin label looks like this, and it gets attached via cysteine chemistry, but click chemistry or even non-natural amino acids work as well. And the fourth group of applications is looking at short-lived radicals that are involved in biological processes. So for example, oxygen reactive species that are involved in oxidative stress, in DNA damaging, but also in wound healing. This is one example, and EPR is more or less one of the only methods you can use to track these species. So, okay, now we make use in EPR of the fact that our unpaired electron can interact with a magnetic field and with electromagnetic radiation. And in EPR, this is radiation in the microwave range. And for Expand, like I'm going to present today, that's around 9.5 megahertz. The simplest system is a free electron zooming around. If our free electron with its magnetic moment interacts with a magnetic field, it can orient either parallel to the magnetic field or anti-parallel to the magnetic field, resulting in two energy levels. Low level is parallel, high level is anti-parallel. And the energy gap is dependent on the G factor. The G factor is a compound characteristic, pretty similar to the chemical shift in NMR, and it's dependent on the field. So what we do in EPR is we scan the field and keep the electromagnetic radiation constant. And if the energy of the electromagnetic radiation matches the energy gap induced by the field, we get absorbance. And for reasons that help optimize signal to noise, the recorded spectrum in EPR does not look like an absorption spectrum, but it looks like the first derivative. So we need a magnet, we need a radiation source. That means, oh, I'm actually not quite there, sorry. Getting ahead of myself. Our free electron usually isn't really free because it's in an atom or in a molecule, so it has neighboring nuclei it can interact with. So if the neighboring nucleus actually has a nuclear spin, so that means it also has a magnetic moment, that nucleus induces a magnetic field. So now our electron can orient itself with respect to the outer field and with respect to the field of the neighboring nucleus. In the simplest instance, the neighboring nucleus would be a hydrogen with a magnetic spin quantum number of 1/2. And that would mean both our levels get split one more time. There are 2 allowed transitions, so our spectrum would have a 2-line spectrum. And in fact, the number of lines in an EPR spectrum can be calculated by looking at the magnetic spin of the interacting nucleus, and it's calculated 2 times the magnetic spin plus 1. So that means interaction with a hydrogen, magnetic spin 1/2, makes 2 lines. Interactions with a nitrogen, magnetic spin 1, makes 3 lines. Magnesium 5/2 makes 6 lines, and so on and so forth. Okay, so we need a magnet and we need radiation. That means we need an instrument. So in both our facilities, ZIP and ARC, what we have is a continuous wave Alexis 2 E500 spectrometer from Bruker. It consists out of the magnet, then the bridge. In the bridge, the microwave radiation is generated, and also the detection takes place in the bridge, and a computer and control elements, of course. What we also need is a resonator. The resonator is between the magnetic poles. The electromagnetic radiation gets coupled into the resonator, and our sample goes in here. So in both facilities for X-band, we have a super high resolution resonator that allows us to do experiments with extremely low number of spins. In our case, instead of a front plate like this, we actually have a grid so that we can couple light in to see light-induced reactions. In ARC, you guys have, in addition for X-band, a cylindrical resonator like this. Those are optimized to do experiments in aqueous solution using flat cells. And you have a dual mode resonator, which is optimized and designed to tease out allowed and forbidden transitions. For example, if you look at triplets or certain transition state metals or the B radicals. And in addition, you guys have an L-band resonator that's, if I understood this right, brand new. And that goes together with a different bridge, L-band, because L-band is lower energy than X-band. And that can be an advantage if you're working with samples that have a high dielectric constant. Now in ZIP, we have, in addition to the high sensitivity resonator, classic rectangular resonator. This is a workhorse. It can do a lot of things, but its sensitivity is considerably lower than for the SHQE resonator. And more important for the work I'm talking about today, we have an X-band loop gap resonator. Those are designed for work with aqueous solutions, and it allows us to do experiments with volumes as low as 5 microliters and a concentration range, spin range, 20 to 200 micromolar, depending on the application. In addition, you have a temperature control system, and I really envy you about that, which is a closed circle helium cryostat that can go down to 4K. Our system works with liquid nitrogen and nitrogen gas, and it only can go down to 100K, but theoretically it can go up to 500K. I have never tried that out yet. Okay, so let's dive in. And I want to start diving in by looking at site-directed spin labeling, because in my, granted, slightly biased opinion, this is one of the strongest method for investigating biological macromolecules at room temperature using basic X-band CV EPR. So why site-directed spin labeling? Well, you can actually use it without having a native paramagnetic center, and you can use it on all kinds of, absolutely all kinds of biomolecules. It works on soluble proteins, well-folded proteins, but also intrinsically disordered proteins, Molten globe structures. It works on membrane proteins. It works on membrane proteins associated into artificial membranes, into even in their own native membrane. So this is pretty good. It works on RNA, DNA, lipids, complexes that can be so as complex and big and complicated as you want. It works on amyloid fibers, basically anything biological you can think of and put a spin on. And the reason for this is because in EPR, you have no size restriction. So if something is big, it's not a problem like it might be in NMR, for example. The sample can be light scattering, which doesn't work with most other spectroscopical methods. The sample doesn't even have to be soluble. That's the reason why it works with amyloids, for example. The sample size is relatively small, as I already said, and it can be done in nearly any buffer and solvent, with the one exception, no reducing agents, because they would reduce your free electron and you would lose your signal. The information you can get are amazing, I think. You can get information about secondary and tertiary structure, about conformational changes, structural dynamics. You can probe interaction interfaces. You can figure out what parts of your membrane protein are inserted in the membrane, how deep they're inserted in the membrane, in what configuration they're inserted in the membrane. You will see that in a little bit. And you can even measure KDs in some cases. And the magic is in spectral line shape analysis. So if you look at this, this is our nitroxide spin label. And if this guy is rotating free in solution as fast as it can, you get an isotropic 3-line spectrum because our electron interacts with the nitrogen. And the signals are pretty sharp. Now, if you slow this guy down, the slower it moves, the more the line shape changes. You can see the line shape here in the extreme. This is a powder spectrum, is completely asymmetric. You can see that with decreasing rotation correlation time, the center line width here gets wider, and the low and the high field peaks slowly move out and also get wider. And this is something we make use of if our spin label is now attached to a big protein, where the apparent rotation correlation time of our spin label has contributions from the whole protein rotating, the backbone fluctuations where it's attached, and from rotations around these bonds. Now for EPR, we actually take this out of the equation by making the whole thing tumble so slow that it's not visible on the EPR timescale. And that would be a rotation correlation time of more than 20 nanoseconds. For big complexes and proteins, that's not a problem. For smaller ones, people usually add either 30% sucrose or 25% phycol to make it tumble slower. And at that concentration, it does not affect backbone fluctuations or rotations around bonds. So what does this mean in praxis? So let's have a look. So this is a slide I stole from the Hubble lab. If you look at the spectra here, in this instance, the spin label is attached to a very flexible loop. So you have big contributions from backbone fluctuations, and this can rotate freely around its bonds. And so this gives you this only slightly immobilized looking spectrum. Now, if you instead of that, move and set the spin label here on an alpha helix, but surface exposed, it can still freely turn around here, but the backbone movement is strongly restricted because of the alpha helix. And as you can see, that spectrum starts to broaden. You can see that the high and low field peaks move out, and that's probably a little harder to see. The peak to peak distance here is also getting slower, wider. The next position is sitting here on the tertiary interaction side, where the backbone movement is restricted, and now it's sandwiched between two interacting alpha helices, so it can't rotate freely anymore. And as you see, you get an even broader spectrum. And spin labels are actually very well tolerated and very rarely mess up folding or activity. And so you can often get it right smack in the middle of an alpha helic abundance, somewhere where it's buried in the structure. And if that happens, you get the broadest splitting here. And this is something that starts to approach what the powder spectrum you saw earlier would look like. So this does not only hold true for these four positions. Guys, I have a little problem. My slide doesn't advance. Let me stop sharing for a moment.
Yeah, no problem.
Okay. And then let me start sharing again. It's working again. No idea what this was, but I am back. Okay, so it actually holds true for basically the whole T4 lysozyme, which is what we just looked at. And it's so you can see that loop regions, helical surfaces, tertiary contacts, and buried site nicely cluster. And this is true for, and it's not advancing again. This is true not only for T4 lysozyme, it has been done for quite a few proteins from different groups. And as you can see, this is a nice way to get a first idea about tertiary structure. But tertiary structure idea doesn't help you too much if you don't know anything about the secondary structure. And just by looking at mobility and just by looking at the peak-to-peak distance of the central line, the one that broadens slowly, you can actually do that by using a nitroxide scan. So you would make mutants in each of these positions following an amino acid sequence. And for example, here in the case of a beta strand, you get this up and down, up and down, up and down pattern if you plot the inverse of the central line width against the amino acid number, up and down, up and down. And this is because going up, it's mobile because it's surface exposed, going down, restricted, mobile, restricted, mobile. And the same works for alpha helices. And you get this more wave-like pattern as it follows the alpha helix. You get an even more detailed picture about what's going on at the single positions if you go through the trouble to actually fit the EPR lines shape using microscopic art and macroscopic disorder theory. And that gives you an idea about actual backbone motions if they are in the pico to nanosecond timescale. You get the apparent rotation correlation time and an ordering factor between 0 and 1 that tells you how good your how extreme the spin label is packed. And if you have multiple populations, you can actually even quantify the percentage of populations. And to a certain extent, you get an idea about exchange rates if they are in the nanosecond timescale. So what this looks like, here's an example. This is myoglobin, labeled in position 135, which is an exposed loop. And you can see here in black the wild type spectrum, and it's an exposed loop, and the spectrum clearly shows this. If you do the fit, so the fit is the dotted line underneath here, it gives you an apparent rotation correlation time of 1.6 and a really low ordering factor, indicating that this is moving around as freely as possible. Now, there is a mutant, a known mutant of myoglobin that has these two residues exchanged against alanine. So you have a big to small mutation opening up a cavity here. And surprisingly, if you label this in the same position and look at it, it actually shows, as you can see in the green line here, a strong population of spin label that is very immobilized. And that comes from the spin label actually getting trapped in this cavity. So this new population in this spectrum has a long rotation correlation time, a really high ordering factor, and nearly 93% of the spins are in this formation. Now it is known that this mutant binds the anesthetic halothen. And if you add halothen to it, you can see that in the spectrum, the populations are now shifted back to the mobile population, with 60% being mobile again. And that is because the anesthetic competes with the spin label for the binding in this cavity. And this can even be used to measure binding in this case by simply plotting the fraction of mobile components against the increasing concentration of the anesthetic. So those are ideas about structure. Now let's talk about looking at membrane proteins. And the key here is power saturation. It is the power of power saturation. Let me first explain quickly how power saturation works. If you look at the center line in your spectrum, the amplitude of that center line depends on the power of the electromagnetic radiation. And that's kind of intuitively to understand, because what we are doing is we excite from a low energy state to a high energy state, and the more power we put in, the more we get, the more excitation we have, the bigger our signal is. But that only works until you start to reach saturation. If you start to reach saturation, what you see is that instead of having a linear increase, you actually start losing amplitude in the end. And when saturation effects start to appear depends on how fast the system can relax back so that you can excite it again. So the idea in power saturation for membranes is you add a paramagnetic collider, either nickel or oxygen. And if a paramagnetic collider collides with our spin label, we can actually exchange spins. So you have a new fast way of relaxation, which means you can put more power in before you reach saturation. And this is what you see here. The white line shows you the power saturation in the presence of oxygen. So there is a collider, and you can see you can put in much more power before you see the saturation effects than if you do this after exchanging all the oxygen against nitrogen in your solution, because nitrogen isn't paramagnetic, so it can't exchange spins. You see it earlier. And this is important. Oh, yes. And by doing those two, you can actually compute an accessibility factor for the collider you're using that tells you how accessible your spin label is for the collider. And this is important in the context of a membrane because oxygen concentrates in the inner part of the membrane much higher than outside in the aqueous solution, while nickel that is usually added as a big complex can't really diffuse in, so you have a high concentration outside and low concentration inside. So if your spin label is attached to a position of your membrane protein that is in the aqueous phase, then nickel will do the best job. And if it's attached in a position that it's inserted into the membrane, then oxygen will do the best job. And that will help you, for example, to do something like this. So this is looking at an alpha helix. And if the alpha helix is in a water-soluble protein, both accessibility parameters, oxygen and nickel, oscillate in phase. Now, if your alpha helix is absorbed on top of a membrane, where it has on this side more oxygen and on this side more nickel, they will actually oscillate out of phase, telling you which end is where. And here in the example is a transmembrane water-filled pore. And you can see they still oscillate somewhat out of phase, but there is also a modulation in the oxygen that kind of tells you where the middle of your pore is, which is pretty cool to see. You can take this a step further, and this is something one of the groups actually here in biochemistry is a pioneer in the method, is if you do both accessibility parameters, oxygen and nickel, you can compute a depth parameter, which you can calibrate using lipids that have spin labels in fixed positions so that you know how deep they're in the membrane. And by doing so, what they do is look at membrane binding proteins, and by assigning depth parameter, they can know that depth parameter that are strongly positive are deep inside the hydrogen carbon core, while the intermediate depth parameters are where the protein interacts with the head group region, and the strongly negative ones is where the protein is in the cytosol. And in this case, for example, they have used this to shed a little light on the activation mechanism for the C2 domain of PKC alpha. Proteinase kinase gets activated by binding calcium and the second messenger lipid PIP2. It can bind to membranes by simply binding to PS lipids. And if it does that, this is the conformation it takes. These loops, these interaction sites are interacting with the head group regions that we can see from the depth parameter. And once one of the PSs is replaced by PIP2, the plane of the body of the protein gets tilted by 40 degrees. This loop that was interacting with the membrane is now completely free, while this loop is now extended into the membrane. And that is most likely one of the components that are important to activate the protein kinase. So this is an example for what you can do looking at membrane proteins. The last thing about spin label I wanted to talk about is measuring distances. And I'm actually pretty excited about this because you can actually really do this with room temperature continuous wave X-band EPR, and it's relatively easy. It is easy because the two spin labels can be actually the same spin labels. So in contrast to FRET in fluorescence, you don't have to worry about having two labels, different labels that need to go onto your protein, and they need to have equal reactivity so that they are not biased on which side they bind and all that kind of thing. It doesn't matter because you're using the same spin label. And you can probe distances at room temperature and with continuous wave between 8 angstroms and 25, but 25, granted, is a little bit pushing it. And again, the magic is in doing line shape analysis. Let's say our two spin labels are attached to flexible loops far away from each other so that they don't interact. So the spectrum of that doubly labeled spectrum would look like a simple addition of the two single spectrums. And it would look like a nice mobile spectrum like we've seen it when we looked at tertiary structure. But the closer the two spin labels come, the more likely they are to have dipolar interactions. And dipolar interactions lead to extreme broadening of the spectra if you get to really close distances. And this broadening you can fit. And by fitting this, you get distance distributions. So this is an example from an older paper, one of the very first working out this method. This was done following this alpha helix, and in the extremes, those two positions would be closest to each other, pointing into the same direction. And you can see here, the black line is the measured interacting spectrum, while the dotted line is the spectrum of the two singles. That is what it would look like if it doesn't interact. And by fitting this, they came to a spin-spin distance of about 6 to 7 angstroms. If they do the same thing using the two positions that are furthest apart from each other, you can first of all see it by eye. It's actually pretty hard to see the difference, but it's still there and you can fit it, and you get a broader distribution between 20 and 25 angstroms. So that was a proof of principle. Here's an application. So what we did here is we looked at the ATP-dependent conformational change of the peptide binding site of the heat shock protein DNAK. So this is the structure of the peptide binding domain. In red, the peptide, and you can see that this slit light-like part is tightly closed over the binding site. And it is known that if you bind ATP to the adjacent ATP binding domain, the peptide gets released. So what we did here is we introduced two cysteines close to each other across the interface, and then measured the spectrum in the spin-labeled spectrum in the absence and in the presence of ATP. And as you can see, in the absence of ATP, there's a real difference between the measured interacting spectrum and the sum of the two singly labeled spectrum, indicating a strong interaction. Here you can see the fit that leads to the distance distributions you see here. And this has actually three populations: a tightly closed one, probably reflecting exactly the structure, one that is kind of intermediate closed, so the lid is a little bit up, and a very tiny fraction where the spins are so far away from each other that they are not interacting. This picture changes dramatically if you add ATP. So if you do the same experiment, adding ATP and do the fit of the interacting spectrum, you get a huge population now where the spins are so far apart that they don't interact, probably reflecting the fully open structure, and a smaller population of the, let's call it halfway open structure. This was pretty interesting for us because it shows that at least in solution, in contrast to the crystal structure, the binding site does sample different different populations, one of them being at least slightly open. And that is interesting because it is known that DNAK can also bind things that are not completely unfolded. It also binds folded structures, and for that the lid does need to open a little bit to accommodate that. So this is pretty nice to see. Okay, so enough about spin labeling, but I want to stay with the nitroxides. So let's go to spin trapping. Spin trapping is important to monitor a host of small radical species that are part of biological processes. The most prominent example would be reactive oxygen species like superoxide, hydroxyl, and a bunch of others that are important because they can actually be quite damaging. So it's important to monitor them and figure out what they do. The problem with them is that they are actually usually not there in high concentrations. At least that's what we hope. They are not very stable, have very fast relaxation times. So even if they are there in higher concentrations, we still can't see them on a normal EPR timescale for a continuous wave expand EPR at room temperature so that we are close to biological conditions. So the idea with spin trapping is that you have an EPR silent component, and those are mostly nitroso or nitron traps that reacts with the short-lived radical, forming a nitroxo radical that is now pretty stable, and you can track it over time. Here are examples for spin traps. I want to point out DNPO, especially because this is the most widely used spin trap. And you get now information about with what radical the spin trap reacted to form the spin adduct by characteristic line shapes. So for DNPO, for example, if it reacts with a hydroxyl and forms this adduct, you get a characteristic 4-line spectrum. On the other hand, if it reacts with a CH3 radical, you get a characteristic 6-line spectrum. If it reacts with superoxide, you get this a little bit more complicated spectrum. And so now let's look at a textbook example that is actually often used for people who want to monitor superoxide radicals to standardize their method. And the system is the oxidation of hypoxanthine to uric acid by xanthine oxidase. it's a two-step reaction that produces superoxide. And if you do this reaction in the presence of DMPO, what you will record is a spectrum that looks like the black spectrum here. And the spectrum can be simulated by a mix of two DMPO adducts. One is the DMPO-OOH. So this is what you would expect if you have pure superoxide plus DMPO-OH, which is the adduct formed with reaction with a hydroxon. Now, the software that comes with the EPR instrument that both our facilities have is able to do 2D experiments where you take spectra at fixed points in time again and again, so that you can monitor a reaction over time. The software is also able to help you fit each of these spectra to figure out the extent of the two components. And by doing the fits, you can actually quantify, count the spins, and know the concentration of both adducts at every point in time during this experiment. So you are able to measure the experiment over time and follow the kinetics. Now let's look at a few examples that I found were pretty interesting. In this example, and that's a slightly older work, the authors looked at the appearance of hydroxyl radicals in isolated rat hearts after they restricted the flow into the hearts for 30 minutes and then removed the restriction and reperfused the heart. And reperfused the heart while they also added DMPO. And what they could see is that in the heart under these conditions, after about 3 minutes, they saw a spike of hydroxyl radicals. And they only saw this in the fraction of the hearts that after this procedure developed heart rhythm problems. And that is important because at this time, at this point in time, it was noticed in the clinical environment that a lot of after patients were released, after they had restrictions of the blood flow in the heart arteries removed, unfortunately, a small percentage of patients would, right in relatively short time, still die from cardiac problems. But autopsies showed that these problems were not the clot coming back or a heart an infarct or something like that, but that it actually came from extreme heart rhythm problems. And so the question is now if these heart rhythm problems have something to do with the hydroxyl radical that were monitored here. And in this work, they actually drove it a step further and they added the moment where they started not reperfusion, so normal flow, they added a hydroxyl scavenger. And then they couldn't monitor, of course, hydroxyl DMPO adducts anymore because they didn't occur. But they also noticed that the likelihood of the hearts to develop cardiac rhythm problems was lower. So this is, of course, isolated rat hearts, and that doesn't mean the same thing happens in humans, but it's a good start and it's a hint and it's something to look at. Okay, this is another example that was actually done in our facility. We had a group in engineering that engineered quantum dots for light-induced nanotherapy. So what they did is they took the quantum dots, put them in cell culture of cells that were infected with a bacteria, and then they show line on it. And what they saw is that the bacteria died and the cell culture, so the cell line luckily did not. And so they looked at it with fluorescence and EPR and could figure out that the likely mechanism was that the quantum dots generated superoxide that then killed the bacteria, but the cells were able to withstand that a little better. And then using EPR, they quantified the amount of hydroxyl of superoxide that was generated, and used this to fine-tune their quantum dots to generate different levels of superoxide, so that they could try out what would work best to kill bacteria, but not the actual cells. And the hope is that this, at some point down the line, could be used as an alternative to antibiotics, trying to tackle antibiotic-resistant bacteria on surface-exposed wounds, for example, on your skin. And one last example. So this is actually an in vivo spin trapping. So what the researchers did here is they co-administered sodium formate and the spin trap POBN into the blood of rats. And after an hour and 24 hours, they checked in bile and urine samples for the for spin adducts. And the reason this is interesting is that formate and formic acid are present in many metabolic pathways, but in high concentration, formate is actually acutely toxic. And it turns out that in methanol poisoning, the accumulation of formate is the main pathway of toxicity. So what they wanted to test is there a radical involved in this? And by using the spin trap and simulating the spectrum they found and doing a couple of necessary controls, they were able to show that, yes, they could trap a radical, and it's a CO2 minus radical. And that indicates that with the CO2 minus, that a CO2 minus radical might be part of the pathway how methanol poisoning works. And so now we know more, and maybe there is a way to actually counteract that by counteracting the radical. Okay, so this is where I actually planned on stopping for today. I know in my abstract, I promised you I would talk about metal proteins and I would talk about intrinsic radicals. But when I put this talk together, I realized that if I do that, I cannot go into any of those methods to a point that the talk would actually be pretty useless. So I thought I'll go into what I know best in more detail, and hopefully have convinced you that EPR spectroscopy, even if it's as basic as a continuous wave and X-band, is a very powerful method to look at biomolecules, and it's really worth considering. And if you want to think about it, and if you have questions, or if you want to toss around ideas, including metalloproteins and intrinsic centers, then talk to me, because if you give me a chance to talk about EPR, I will. So I want to finish this off with acknowledging my funding, of course. I can't say this loud enough. The shared instrument core with over 40 instruments is highly subsidized by the Department of Biochemistry. I was lucky enough over the last 3 years to get 3 grants through the CU Boulder Core Facility Assistant Grant Program that allowed me to modernize certain things in both cores. And I was even more lucky, and I got an R24 just a few weeks ago that now allows me to buy 3 new centrifuges and replace some of our old, over 30 years old dinosaurs that we have in the core. A big thank you goes to Emily. Emily is the PRA that works a third of her time with me and and 2/3 of her time supporting the cell culture facility. And since I have over 40 instruments and an average of about 130 users a year, I absolutely would not be able to do this without her. With this, I want to stop and thank you for your attention, and I hope you're going to have fun with EPR. And so that I finally get to see you all, I'm going to stop sharing now. Yes. Okay. So if you have questions, feel free to unmute and just ask, I guess, right?
Yes, absolutely. I have one question, well, a couple questions from one attendee in the chat, but I would like to just see if anybody would like to unmute and ask any specific questions of Annette. Okay, so the questions that I have in the chat, one of them says, and the questions came through on the slide regarding the rat hearts study. And so I wasn't sure if the question was specific to that slide or more in general, but part one is how to distinguish signals for multiple spin labels attached with a protein in both flexible as well as restricted regions.
Okay, so I think this had nothing to do with the rat heart, actually.
Yeah, now that I've read that for sure, yeah. How can EPR be regioselective, I guess, is the second part to that question.
Right. So, okay, so if you attach multiple spin labels, you do this to measure spin-spin distances. So then you attach 2 labels. If you attach 2 labels to monitor conformational changes, for example, you have an educated guess where you actually put your labels. So you know beforehand that this is a flexible region and this is a not so flexible region, especially also because if you want to do spin-spin measurements, you also have to measure the singly labeled. So you know, you get that information beforehand as well. The second part, what are the possibilities for conformational rigid structures like polysaccharides? So you actually managed to find a biological sample, a polysaccharide, where I'm actually not sure if there are methods to attach a spin label to at the moment. I'm pretty sure a good organic chemist can do it. And the lab where I did my PhD, we did a lot of organic chemistry developing spin labels. So I'm pretty sure it's possible. I think one of the possibilities, again, here would be to measure spin-spin distances so that you get an idea how your sugars are oriented. For example, that's something I could think of. If the polysaccharides are involved in any interactions, you could see changes in mobility, because even if the polysaccharide itself is very rigid, the label would still be able to rotate around its own bonds, and that could potentially be affected if something binds to it. So yes, I think there are possibilities there too.
Awesome. Well, I don't know if there's any other questions. I don't see any in the chat, but if you
I
don't
got
want
one, Brian.
to, okay, yes, go ahead.
Sorry
Corey.
to interrupt.
No.
Mine actually sort of reflects the first question from the chat. So
Okay.
how do you, can you specifically target a particular amino acid residue with your spin label? And how do you do that?
Okay, yes, so there are a couple of methods. So the most common method is actually that people go ahead and in the position they want to probe, they mutate against the cysteine and then label the cysteine there.
Okay.
Another method would be to still involving at some point mutation, but using click chemistry instead. Another method that is pretty new and is being worked out as we speak, so to say, is to use unnatural amino acids, and then label those. And they are actually working out ways at the moment to do this inside cells. First studies doing this are out so that you can actually look at stuff in their native environment. It's not quite that easy because inside a cell, you often have a reducing environment. and that reduces your free electron. But at the moment, there are first spin labels with so big groups around them that they actually shield the spin, and that's pretty exciting.
So all of the methods are, it's mutation based first, so site directed. Yeah. Yep.
Yes.
Yeah.
And I mean that is the weakness, or let's say that this is the
it's a challenge. Yeah,
it's a challenge because especially if you want to look at secondary structure, tertiary structure, build up a structure, you're talking easily about hundreds of mutations.
Yeah, I was thinking about some of those figures that you showed earlier where you had several sites along an alpha helix, and that each one of those is an independent mutation. That's a lot of effort.
That's a lot of effort, but once you so this was never published, and that had completely different reasons. But we tried to look at the structure of TTR in amyloids compared to the structure in solution by doing exactly that. And once you've worked out your mutation strategy, you can actually streamline this pretty well.
Mm-hmm.
And the good thing is that the spin labels are very well tolerated. Mutations to cysteines usually are very well tolerated, and then adding the spin label very rarely messes up folding or structure. It doesn't mean you don't have to test all your mutants to be on the safe side, but your chances of success are very, very high.
Mm-hmm. How long do the measurements take? Like, what is the acquisition time for, say, one protein with a site, spin labeled, site directed?
So that depends a little bit on your application. So if you just want to have a spectrum and you can concentrate your protein reasonably well, let's say 15 micromolar or something like that, then I would say pure acquisition time, maybe 5 minutes. Mm-hmm. If you do a power saturation experiment, you are at it for hours. That's just the truth, and it's painful, but it's worth it. If you do a spin-spin interaction, you have the problem that you get this extreme line broadening, right? And that is bad for your signal to noise. So usually doing those, you have to average more scans. So I would say half an hour to an hour. Okay. Those are typical values.
Yeah, I just wanted to get an idea of what that looked like. Yeah, right. Thank you.
But you have to add loading your capillary and cleaning your capillary in between, which also adds a little bit of time.
Thank you.
Awesome. Is there anybody else that would either like to unmute and ask a question, or feel free to type in the chat?
I have a question about a technical question mostly, as you're talking about the time thing. So one of the slides you showed that from that rat herd, that 30 seconds, then 100. So how is it a continuous measurement, or it's just how would you transfer and do it technically in the instrument?
Right. So for this experiment, what they describe in their methods is that they took the samples and snap froze them until they could then very fast measure one after the other. And you saw the signal of to noise in those spectra was pretty bad. So they didn't take the time to average so that they could really do it like this.
Like a continuous measurement type.
Yes. But there are actually, Bruker makes a resonator that has that is specific for a flow cell, so that you can do basically like a continuous flow experiment. It's a little bit tricky because those things tend to vibrate, and vibration is, as you know, not the best if you want to tune your EPR, but it's totally doable. And also what people have done, especially with metalloproteins, where you mainly work at low temperatures, and low temperatures more like what you can reach, like 30K or so, not what I can reach with our setup. They actually do a setup like a chemical quenched flow. And instead of a chemical quench, they shoot their reaction into an aging loop, and from there into liquid nitrogen to have it snap frozen. and then from there transfer it directly into the EPR. And they can get time resolutions in the millisecond. So that's pretty cool.
That's good. Yeah.
So we just had one more question pop up in the chat. It says, I would like to know if this would be helpful for determining singlet and triplet chlorophyll states and isolated photosystems or biocolloids. Seems like it could be based on your equipment descriptions near the beginning.
Right. I have to be totally honest here. I want to say yes. I'm pretty sure this probably has to be done at low temperatures. I would have to look a little bit into it, but I think, yes, it's possible. And it's very likely that if it's done, then it should be done with your system at really low temperatures. Okay, is that a publication that?
In a really quick Google search, yes.
Yes, thank you.
Oh, yes. Thanks, Corey. Andrew, I think if you have not seen that publication, that might go into a little bit more detail about your question. So
Yep.
definitely speak to us. I mean, Indrani and Annette, if you have more technical questions, but seems like we probably could do those types of measurements
Yep.
on our EPR here.
I'm pretty sure. Yeah, I just had a look. They're doing this at 77K. Yeah. So
Gotcha.
it's borderline between our two. It's better on yours.
Absolutely. Any other questions, please? If you don't want to unmute, feel free to type it in. But any other questions while we have Annette here? All right. Seems
Okay.
like not. So just want to thank you again, Annette, for taking the time and giving the seminar today. We really appreciate it. And sounds like several, if not all of us, will come by tomorrow to check out your facility as well.
I'm looking forward to see you.
Thank you.
Thanks, Annette.
Thank you for having me.
Great
Thank
talk.
you.
Bye.
Bye.
So I hope that was a little bit in the right direction, what you wanted to hear.
Absolutely. Yeah, I
Yeah.
thought that was great. Yeah, very good.
I was
I
gonna
feel like
ask
I
you
will
what's
have
on your
thousands of questions.
I'm sorry?
Like I will have, like, because it's not like I have a lot of experience with EPR, but I probably will have thousands of questions when I have like the how to do stuff for then, like, data part. I understand because my
Okay.
background is like molecular magnetism. So
Mhm.
it's kind of, yes, I understand spin interaction, secondary, tertiary interaction, but how to do the experiment is like where I'm
Yeah.
not very.
If I can help, I'm happy to. And I know how you feel. I took over the X-ray facility with no background whatsoever.
Annette,
Yes.
what's on your wish list? Are you trying to add new things to your EPR facility, or?
No, we actually just added something, and so we actually bought. So Caroline bought the mass photometer.
Mm.
So we have now this, and I'm working out methods for that at the moment.
Mhm.
And it's a really, really cool method to